Okay, the second talk will be by Dr. Keji Zhao from the National Heart, Lung, Blood Institute. He's the Director of Systems Biology Center, and the title of the talk is Trac-looping, a novel technique for analysis of chromatin accessibility and interaction.
Thank you for the introduction. And I'm going to tell you our recent method called trac-looping, and it's a new method for chromatin, for analysis of chromatin accessibility and the interaction. And as you all know that DNA transposition is mediated by DNA transposes such as TIN5, which recognize the mosaic ends sequence of a DNA transposer, and then form a circular dimeric complex. So this complex can be inserted into the genome, resulting in DNA transposition. And however, the TIN5 enzyme can bind to a monovalent or one single ME sequence to form this dimeric TIN5 ME complex without the non-spacer region. So this dimeric complex can also be inserted into the genome, but instead of transposition, then it results in DNA fragmentation, and which is the method actually used by ATAC-seq. So in order to capture chromatin interaction in the genome, we designed a bivalent linker for trac-looping. So this bivalent linker contains two mosaic end sequences separated by a short oligonucleotide linker, and then addition of TIN5 enzyme into this bivalent linker, it forms a tetrameric complex like this one. And this is the region we use for detecting chromatin interaction. So this work in my lab was done by, the experimental work was done by Qingzhong Tang, and the data analysis was performed by Bingbing Lei, Wenfei Jin, and Gangqing Hu. So how this works? So for the trac-looping experiments, and we add this tetrameric bivalent complex into formaldehyde fixed cells, and then this complex, one end of this tetrameric complex will be inserted into one chromatin region, and then the other end will be inserted into another region. So after insertion, and then the genomic DNA is cleaved into small fragments using ELA3, and the region containing this bivalent adapter sequence was purified using a biotin streptavidin system, and then it is amplified for paired end sequencing to generate the library, basically. So the chromatin actually is organized into multiple different structures in the genome. It's a very heterogeneous structure, and some regions are nucleosome-free, and therefore it's accessible. Other regions are highly folded, and then there are even other regions that contains promoter-enhancing interactions. So this tetrameric bivalent complex can be inserted into different regions, chromatin, therefore reveal different information for chromatin organization. So first of all, for example, when it's inserted into the nucleosome-free region, it will generate the complex that is the PETS that is smaller than 150 base pairs, revealing chromatin accessibility. And for other regions, for example, once inserted into this high folded region, it will generate DNA PETS that's spaced by between 150 base pairs and 1 kb. This reveals the folding pattern in the genome. And then if the complex inserted into the region with the promoter enhancing interaction, then it can reveal produce PETS longer than 1,000 base pairs, which reveals information for promoter enhancing interactions. And so to test how this trac-looping works, we purified human primary CD4 T cells from human blood, and then perform this assay. And then here are 4 experiments we did. As you can see that we obtained about 400 million PETs from each library, but for unique PETs, we get only 50 to 120 million, depending on different experiments. So the distribution looks like we get from 30% to 60% of PETs that's smaller than 150 base pairs, and 20 to 25% of PETs between 150 and 1,000 base pairs, and then about 20 to 30% PETs longer than 1 kb. So next, I'm going to walk through each of these different categories of PETs to see how they reflect or reveals information for the genome's chromatin structure. So first of all, PETs smaller than 150 base pairs. So this is just genome browser image in comparison with the ataxic PETs, fixed cells or affixed cells. As you can see that the Trac-looping PETs are highly similar to the ataxic PETs, and therefore the Trac-looping PETs smaller than 150 base pairs detects chromatin accessibility. So the next category is the PETs between 150 and 1,000 base pairs. As you know that the chromatin fiber can be folded in 2 potential configurations. One is called a solenoid, like this pattern, and the other is a zigzag folding pattern, like this. So if it's a solenoid, like this is an N1 nucleosome, then it's next to especially with an N5 or N6. Therefore, insertion of the bivalent linker between the linker at this position at N5 or N6, this will generate a PET separated by 4 or 5 nucleosomes. But if it's a zigzag pattern, then insertion between N1 and N2, and the other end of bivalent complex inserted between N4 or N5, therefore resulting PETs separated by 2 or 3 nucleosomes. So what we did we see then in the genome? So this is the PET distance. The blue line represents the PETs for all accessible regions. As you can see that we did observe peaks with N+2 nucleosomes or N+3 nucleosomes. Therefore, this corresponding to the zigzag chromatin folding pattern. But the blue line shows very little enrichment with N+6 or N+7, indicating that there's a very small fraction of chromatin that's folded by in the solenoid configuration. So in accessible regions, it's mainly zigzag folding, but not solenoid. But for the inaccessible regions, represented by the pink line, we detect peaks both at N+2 and also N+5 or 6, indicating the existence of both zigzag and solenoid pattern. But for inaccessible chromatin regions, that can be associated with either K9 methylation or H3 K27 methylation. So looking at the K9 methylation, then we see very little N5 or N+6, suggesting that the K9 methylation region mainly is zigzag, but not solenoid. But for the K27 methylated regions represented by the yellow line, then we see peaks at both N+2 and N+5 or 6, representing chromatin folding for both zigzag and solenoid patterns. So next, pre-studies show that chromatin interaction domains are separated at active promoters or GTCF binding sites, mainly at a resolution of 10 kb to 20 kb. And we want to see if we can observe this at higher resolution. So here we used 10 base pair resolution. And so this is on the top track panel that shows the Trac-looping data. And this shows the silent promoters. This is RTKM value of gene expression. You see that there's not much separation between upstream or downstream of TSS, but for active promoters, and we see strong interaction on each side of TSS, but very few of them across this TSS. You see in the middle. And the similar thing was with CTF. So on one side, we see strong interaction. The other side, also strong interaction, but very few cross this TSS, CTF binding site. But in control, for DHS sites without CTF binding, we see the interaction intensity on each side and also across the site are similar, so there's not much clear separation. So the lower track panel shows the high C control. As you can see that we don't see much separation based on this at 10 base pair resolution. So the next category is PETS, Trac-looping PETS, from 1 kb to 200 kb. And you can see that at the peaks of H3K4 methylation representing promoters, or peaks at the K27 acetylation representing enhancers. So high C PETS not enriched as expected, but both the GR PETS and Trac-looping PETS are highly enriched, indicating that Trac-looping PETS may specifically detect promoter-enhancer interactions. So this is the example of the genome basically containing the TRAP10 gene. As you can see that these are chromatin accessibility represented by shortcuts from Trac-looping, and these are interaction matrix. And as you can see that we indeed observe the interaction with elevated red signals between promoters and also enhancers. And these interactions actually confirmed by the high C data from the same cell or the high C data from the GM12878 cells, although the background is much higher. And these are all the significant interactions detected with the Trac-looping. And so in total, we detected 85,000 significant interactions from a total of 265 million pairs from these libraries. So the question then we want to see is how robust these 85,000 interactions, whether the detection is this is saturated or not. So for this question, we separated these interactions into different categories based on their significance. So the highly significant ones, these are blue and with a dark orange. So these two categories occupies 25% of all interactions, and then the less significant and the least significant. So to test the robustness of these interactions, we downsampled the library and then see how many of them can be reproducibly detected. As you can see from this plot that for the top two categories, and even we used 50% of the PETS, then we can still detect more than 80% of all the interactions, suggesting that indeed these interactions are saturated and can be reproducibly detected. But for the less significant interactions, they are far from being saturated, therefore need more sequencing rates to reproducibly detect them. So to show that Trac-looping detects functional promoter enhancer interactions, we showed one example for the interleukin-2 receptor gene. This is a critical gene for T cell activity and T cell function. And so this track shows the accessibility. So this is the promoter region, and these are potential enhancers. And this is the raw interaction matrix. And here shows all the significant interactions in this region. And this panel shows the interactions directly linked to the promoter for interleukin-2 receptor gene. And all of these interactions actually are validated by the capture-C analysis using the IL-2 receptor promoter as a bait. So for these interactions, we selected 3 potential enhancers to test using Cas9 crab system to see if we can inhibit these enhancers. Actually, indeed, when we used this Cas9 crab system, then we found that we can inhibit this gene expression of interleukin receptor significantly after transfecting of this construct into the cells, suggesting that Trac-looping indeed detects functional promoter enhancer interactions. So next, then we will look at the higher order chromatin organization, such as TAS structure or compartment organization. As you can see, that compared between Trac-looping and in situ high-C, they are highly similar using this interaction matrix. And also this TAS structure shown by this, and also the blue, that's the TAS from the high-C, the red TAS from Trac-looping, they're highly similar. And also for compartment scores, this is a CB compartment score from Trac-looping, and compared with the score from high-C, they are highly correlated. And also they are highly correlated in the boundary scores at y-axis and x-axis here. And furthermore, when we look at the orientation of CTCF motifs from these interactions detected by Trac-looping, we found that CTCF anchored interactions, if it's longer than 200 kb, so the vast majority of the CTCF motifs are in the convergent orientation as found previously. But if it's between 10 kb and 200 kb, the convergent size still dominant, but it's with a lower fraction. But if the interacting regions are within 10 kb, then more or less the motif orientation are random. So next, we asked whether we could detect differential promoter enhancer interactions using Trac-looping. So to test this idea, we isolated the resting T cells and stimulated them using anti-CD3, anti-CD28 antibodies for 24 hours, and then performed Trac-looping on both of these cells. And this is the data. The top tracks shows the chromatin accessibility, and we see some regions show changed accessibility, others don't. And these two tracks showed the interacting matrix. The red circle indicated the size, that showed increased interaction by stimulation, but not increased accessibility. And then the orange circle shows the region that demonstrated both increased interaction and increased chromatin accessibility. So this also confirmed by the high-C analysis, as you can see these regions. So in total, we identified about 3,500 sites showed increased interaction, as shown by the red, and 750 showed decrease in interaction. Then the question for us was, how many of these actually is related with increased chromatin accessibility? So to test this, then we normalized the interaction based on the accessibility. So the y-axis shows the change in interaction, and the x-axis shows the fold change of accessibility. As you can see on the top, the red dots, about 34% of regions that show both increased accessibility and increased interaction. But the vast majority, 66%, that show only increase in interaction, but not in accessibility. So this suggests that chromatin accessibility alone is not sufficient for the chromatin interaction detection by Trac-looping. So by using the 4DN award support, we also generate libraries, libraries for the 4DN cell lines. Two cell lines we used first. That's H1, the ESC, and also the GM file, H7H cells. We generated 4 libraries for each, and each library we generated a sequence about 200 million unique PETS, 200 million PETS, total PETS. And this is just one piece of the information we showed here. So these are the significant interaction detected from this PETS for this is for GM cell line, and this is for the H1 cell line. As you already see from this minimal analysis, that there's a very different interaction pattern between these two cell lines shown by the Trac-looping. So in future, with the 4DN support, we want to further optimize the Trac-looping technique. For example, at the moment, then we use the spacer length for the bivalent linker. We tested only 30 base pairs, and because the chromatin interaction may show different proximity, therefore different spacer lengths may be required to capture different chromatin interaction. Therefore, we are going to test different spacer lengths. And then secondly, we are generating more Trac-looping libraries for the 4DN cell lines, and we also wish to compare the different features of Trac-looping with other interaction techniques. So I'm going to stop here and take any questions.
Okay, thank you for a very nice talk, and we'll tune in again in 1 month.
Okay.